Hi everyone, welcome to the crash course of thyroid metabolic hormones. I'm going to be referring to the book Guyton, so let's dive deep into the details. This is the thyroid gland, which is one of the largest endocrine glands of the body. It sits below the larynx and right in front of the trachea. It has a left lobe, a right lobe, and the central portion is called isthmus. We're not going to be going deep into the anatomy because this video primarily focuses on the physiology of the thyroid gland and its hormones. Thyroid gland secretes two main principal hormones, which is T3 and T4, triiodothyronin and thyroxine. And also the C cells of the thyroid gland, it is capable of secreting another very important hormone called calcitonin. This video focuses on these two hormones. Now T3 is more potent of the two, but thyroxine is released in a greater quantity compared to triiodothyronin. But eventually, all of this thyroxine is converted to triiodothyronin because the major function is performed by T3, which is triiodothyronin. The function of these hormones is to increase the basal metabolic rate. When you talk about the physiological anatomy of the thyroid gland, so here, this diagram, you can see this is the thyroid gland and it has multiple follicular cells. These are the follicular cells. These closed follicles, they are lined by epithelial cells, and these are cuboidal epithelial cells. Cuboidal epithelial cells. And inside these follicle cells, you'll find the colloid, right? So I hope you're getting the bigger picture. This is the thyroid gland. It consists of closed follicles, multiple closed follicles. These closed follicles are lined by cuboidal epithelial cells, and this is a magnification of this cuboidal epithelial cell. And inside the cuboidal epithelial cell, you'll see colloid. Another very important information to take note of is that the thyroid hormones, they're never floating freely, neither in blood nor when they're stored. They're never free. Whenever they're stored in these thyroid follicles, they're always bound to this very important protein called thyroglobulin. So they're bound to thyroglobulin, and that's how they're stored. Wherever they're released into the blood, even then they're not free. They are bound to thyroxine binding globulin. Talking about the production of this hormone, the thyroid hormones, it's important for you to understand that this is the GIT, and in GIT, you have iodide ions. Now, you may be wondering where do these ions come from? So, whatever food that you ingest, it has a certain amount of iodide ions. These come into the blood capillaries, and via the blood capillaries, these iodide ions are taken up by the epithelial cells of these follicles right here. Now, this follicle, it has a basal or plasma membrane, which is next to the blood capillary, and it has an apical membrane, which is basically facing towards the colloid. So right here, everywhere is colloid. I hope you're understanding the bigger picture. Now, right here, you have sodium iodide symporter. Sodium iodide symporter is capable of transferring two sodium inside and also one iodide inside. That's the reason it's called symporter, because both of the ions are transported in the same direction. Now, what happens is that there is a gradient that helps to bring about this change. Before that, let me tell you that sodium concentration is greater outside and it is lower inside. So that's the reason from high concentration to low concentration, sodium is being carried, and this is facilitated diffusion. Whereas for iodide, it is secondary active transport, because iodide ions eventually they become greater in quantity inside, so the quantity inside is higher. That's the reason this is secondary active transport. It is active transport because from low concentration iodide is being pumped to a greater concentration. It's secondary because it derives its energy from a different gradient, and that energy comes from the sodium potassium ATPase pump, which is right here, that pumps 3 sodium out. That's the reason sodium is out. Sodium is high in quantity outside, and 3 sodium out and 2 potassium inside. So this 3 sodium out, this means that easily facilitated diffusion will occur for sodium, and the energy from this gradient helps to pump iodide inside as well. Now this entire phenomena, sodium iodide symporter, this is known as iodide trapping. So we have trapped iodide inside. Very important. Now, this iodide trapping, it is stimulated by a very important hormone called TSH, which is thyroid stimulating hormone, and it is secreted by anterior pituitary. Anterior pituitary releases this TSH, and it comes and it acts on thyroid gland. But what exactly in thyroid gland? So it acts on this very important transporter, sodium iodide importer. So I hope you understood that for now. Now, after iodide trapping, a very important thing happens. Okay, so since I've told you that TSH, it acts on this importer, hyposectomy, which is basically removal of the pituitary gland. The pituitary gland is also referred to as hypophysis. So hyposectomy, the removal of hypophysis, greatly diminishes the activity of the iodide pump in the thyroid gland. So that's one thing that you need to know. Now, once iodide is inside the thyroid cell, the thyroid epithelial cell, you have chloride iodide ion countertransport molecule here, which is known as penrid. What does it do? It pumps the iodide outside, meanwhile pumping the chloride ion inside. So this is basically an chloride iodide countertransport because it is pumping both of them in the opposite direction. Once iodide goes out, now, there's another problem. This iodide cannot be used in this ion form. It needs to be converted to a nascent or a lesser reactive form. For that, another thing, a reaction happens. So, in this apical surface, we have peroxidase and we have hydrogen peroxide. These help with the conversion of iodide into nascent iodide or I3 negative. So, now these both forms are usable forms. Now let's leave this here. Let's go back inside. I told you that there is thyroglobulin, which does what? Which basically helps bind the thyroid hormones T3 and T4, and it keeps them in the stored form inside, right? For now, we have not made T3 and T4, so let's just forget about that. But it's important for you to understand that thyroglobulin exists. And this thyroglobulin molecule, it has multiple tyrosine. Tyrosine is what? It's an amino acid. So there are at least 70 tyrosine amino acids inside. So it means it is capable of holding a lot of thyroid hormones, at least 30 thyroxine and a few tyrothyronin molecules. This one thyroglobulin can hold. And how is this thyroglobulin produced? So inside we have first DNA. So from the DNA, you get mRNA, and that mRNA gets translated to proteins. It goes to endoplasmic reticulum, then it goes to Golgi apparatus, and eventually you get the thyroglobulin. So thyroglobulin is inside. Now, in order for um, thyroid hormone to be formed, it's important for this thyroglobulin molecule to go outside. So this thyroglobulin molecule goes outside, and then what happens is that these two things they come in contact. The iodine, which is now in nascent form, iodine and the thyroglobulin, they come in contact. And this coming in contact of tyrosine or this coming in contact of iodine and thyroglobulin is basically referred to as organification. So now the next thing that will happen will be organification, right? And what was this that happened here? This was oxidation. What was this? This was oxidation where we converted the iodide ion into a neutralized form, right? So we oxidized iodide ions into iodine. And what was the enzyme that was necessary for this? It was peroxidase, and you also needed hydrogen peroxide. Whenever peroxidase system is blocked or when it is hereditarily absent from the cell, the rate of formation of thyroid hormone falls to zero. So there's some cases where this enzyme is absent hereditarily, so thyroid hormone production also falls to zero. After oxidation, very important, we're going to refer to this procedure called organification. What exactly is organification? Organification is when the thyroglobulin molecule it comes in contact with the nascent iodine. When they both come in contact, that procedure is called organification. So iodination of tyrosine and thyroid hormone formation. So where is tyrosine? Tyrosine is inside the thyroglobulin molecule. So iodination of that tyrosine, because this iodine is going to come in contact with the tyrosine. So iodination of that tyrosine, and then ultimately the thyroid hormone formation. These two things are referred to as organification. Or in simple words, you can say that this is basically organification. Iodine plus tyrosine or thyroglobulin that forms thyroid hormone. So that is referred to as organification. Now, what happens is that thyroglobulin is released from the Golgi apparatus where it was produced and through the apical cell membrane into the follicle. This is the follicle, right? Colloid. Colloid is where the organification is going to happen, and this is where now thyroglobulin resides. And there'll be a coupling reaction that will occur. After this, what will happen? There'll be a coupling reaction, and in that coupling reaction, ultimately thyroxine will be formed. So, what is that coupling reaction? Let's look at this. This is the coupling reaction, and this is the entire flow chart from the book. So, the tyrosine that is there, where exactly? In the thyroglobulin, and this is the iodine that we formed. So, this step, the first step, this is referred to as organification. This is organification, and this entire, let me change the color, this entire process, this is coupling. So in this coupling reaction, what happens? First of all, you have tyrosine, then you have a monoiodotyrosine, then you have diiodotyrosine, then you'll have triiodothyronine, you'll have triiodothyronine again. These are the changes in numbers, and then eventually you'll have thyroxine. So what? First of all, tyrosine, then monoiodotyrosine, then diiodotyrosine, then triiodothyronine, and then thyroxine eventually. So coupling reaction happens where you eventually get thyroxine. So that's the reason I was telling you thyroxine is produced in the greatest quantities compared to T3 triiodothyronine. Even triiodothyronine is produced, even RT3, which is triiodothyronine, or you can say reverse T3 is produced, but in greatest quantity, thyroxine is produced. And this is sufficient to supply the body with its normal requirements of thyroid hormones at least for 2 to 3 months. And yeah, once this is formed, what will happen? It has to come inside, right? Inside the follicular cell, and then it will be released into the body. So it's formed here, it has to come inside the cell, and then it will be released into the body. Most of the thyroglobulin hormone is not released into the circulation. Before that, let me go in steps. So the apical surface of thyroid hormone of the thyroid cell, sorry, the apical surface of the thyroid cell, it sends out pseudopod extension that close around the small portion of the colloid. So not only do they take up this entire spaceship right here, which consists of the organified molecule, which is thyroglobulin and iodine, it not only takes up the spaceship, but it also takes up some of the colloid, right? And it forms a pinocytic vesicle. This pinocytic vesicle, it enters through the apical membrane and it goes into the thyroid cell. Once it's in the thyroid cell, it's going to form a vesicle, right? It's going to form a vesicle that has Tg and IO. Now, this vesicle is going to fuse with the lysosomes. They're also lysosome packed vesicles, right? Once these two come in contact, it's going to form a digestive vesicle. What is digestive vesicle? Digestive vesicle basically has Tg, IO, and lysosomes. So basically, both of them have fused together. That's a digestive vesicle. When digestive enzymes they act on this entire spaceship, when the proteases act, what happens is that the T3 and T4 that has been formed, they're released, and even the thyroglobulin, it's released. But this thyroglobulin, it's basically broken down and it is recycled. And what about T3? Even the T3, it's broken down and it's recycled, whereas the T4, it's released into the blood. So this is something that you need to know. And what happens? How is this released out? So some of the thyroglobulin in the colloid enters the thyroid cell by endocytosis after binding to the myelin, a protein located on the lumen membrane of the cells. The myelin thyroglobulin complex is then carried across the cell by the transcytosis to the basolateral membrane. So this is one thing where pinocytic vesicle is formed. First of all, the pseudopod is formed, and then um, pinocytic vesicle is formed. But there's another way. There is myelin. Now this myelin is going to do what? It's going to, you know, bind with the entire spaceship, and it's going to form a myelin thyroglobulin complex, and it's going to bring it inside the cell, and then transcytosis will happen, and that's how it's going to take it out via hair. So transcytosis will happen. So this is the second way of bringing in your thyroglobulin molecule. Eventually, you'll get the thyroid hormone, and thyroid hormone is secreted out. And another very important thing is that these are not secreted into the blood. What happens is that about three-quarters of the iodinated tyrosine in the thyroglobulin never become thyroid hormones, but they remain monoiodotyrosine and diiodotyrosine during the digestion of thyroglobulin molecule to cause release of thyroxine and iodothyronine. These iodinated, I told you that this process it can also stop in the middle, and you'll just have monoiodotyrosine, diiodotyrosine. These never are released. They remain in the follicular cell and they're freed from thyroglobulin molecules, but they're not secreted into the blood. Instead, what happens is that very important iron enzyme called deiodinase comes into function. This deiodinase enzyme it's going to break these down and also the thyroglobulin molecule, and it's going to recycle it and make it again available for the gland to form additional thyroid hormones. And there could be congenital absence of deiodinase enzyme, and it can cause iodine deficiency because of the entire failure to recycle the thyroid or iodine, basically iodide or iodine. So there were two deficiencies that could occur. One was a deficiency of peroxidase, and other was deficiency of deiodinase, which was important to break down these and recycle them. Um, about 93% of the hormones which are released from the thyroid gland are normally thyroxine, and only 7% of triiodothyronine is also released. So, sorry, initially I mentioned that T3 is not released, but T3 is also released, but it's only 7%. 93% of it is T4, and 7% is the 3 T3. Now, moving forward, transport of thyroxine and triiodothyronine to tissues. Now, okay, we'll cover that in the next video. I think this is enough for this.